I'm going to introduce the cell biology lab, looking at differences in gene expression for DNA damage. And we have a really fun group. Justin, Chase, and Alexi are going to present their amazing talk and have a game as well. So have fun.
All right, can everyone hear us all right? Good, cool. My name is Alexi.
I'm Chase.
And I'm Justin.
And we study WD40 gene expression in the presence of DNA damage. This is the 3140 Cell Biology Laboratory at the University of Colorado Boulder.
And just to start things off, we know Zoom can get a little bit boring sometimes. So if you weren't paying attention, now is a great time to pay attention because we have a really fun contest for you with an amazing prize. And this is like a physical prize. So it's not just like a high five, something you want. But basically the contest is we've drawn this diagram of our model organism that we're going to talk about in a minute. This is Tetrahymena thermophila. And if you can give us the correct number of this diagram that's in our presentation when we ask you for it, the first person to give us the correct number will win an amazing prize. So this is number 1 on this slide.
All right, so in this class, we were really looking at what genes are involved in the DNA damage repair pathway, specifically those genes found in our model organism, Tetrahymena thermophila.
And there are a lot of reasons to study DNA damage repair. The most obvious and most important is that it's frequent in all organisms. So for example, when you go outside, a lot of people wear sunscreen to protect themselves from damaged DNA by UV light. And UV light is something that's emitted by the sun, and so it's a super common thing. Damaged DNA can lead to diseases such as cancer or skin cancer in that case, but not always because organisms tend to have good DNA damage repair pathways. And so that's what this class is all about. We're looking at those pathways, what's involved, what's not involved. And then the other thing is that there's the possibility for other discoveries involving DNA. So how is DNA broken? How is it repaired? And general gene editing research.
So for the study, we can't initially start on human cells, so we have to do it on Tetrahymena thermophila. And this is a great model organism because growing it in a lab setting, it's relatively safe and inexpensive, so many trials can be done. It's been extensively researched, and a specific part about it is that it undergoes conjugation, in which when it's reproducing, it cuts up its genome and then fuses with another Tetrahymena cell and fuses with that to make it a whole new genome. And that undergoes a lot of DNA damage repair. And that's why it's such a good model organism. And as you can see on the left, there's a magnified version of it. And on the right, they're just swimming around, being very active, playing with little pieces of sediments, kind of recreating the World Cup.
And so within every one of our model organism, there's a genome, and WD40 is one of those genes. Unfortunately, it's not related to the oil, but we got the oil's logo flag behind us. But there's little known about this gene itself. We know that it codes for a WD40 domain protein, and that WD40 proteins are known to respond to DNA damage. Similar proteins are involved in cell signaling, RNA processing, and cytoskeleton assembly. And this gene is also found in other organisms such as humans, which means that any research that we're doing here could be applied to other organisms. So all of these are important processes, and that made us start to think that it's possible that WD40, this gene, is involved in the DNA damage repair pathway because that's another important process. Another thing that we were looking at before we started research was this gene's expression compared to another gene that we know what it does. So RAD51 is the gene on the left, and these are two expression profiles, RAD51 and WD40 on the right. And expression profiles are basically showing on the y-axis, they're showing the expression level. So how much RNA is produced, how much is this gene actually being expressed? And on the x-axis, this is the time. So we're looking at time during conjugation, which is the time that Tetrahymena is actually cutting up its genome and inducing DNA damage. And notice how both of these graphs are very similar in their spikes. So there's a spike in the beginning and there's a spike at the end. And this made us think that since we know RAD51 is involved in the DNA damage repair pathway, it's possible that WD40 might have a similar role. So all of those data led to our hypothesis that WD40 is involved in the T. thermophila DNA damage repair pathway. To test this hypothesis, we used a bunch of methods to measure gene expression. But the most notable were PCR and gel electrophoresis. Just a brief introduction to PCR, or polymerase chain reaction, is that it's based on the fact that DNA itself is really hard to study. So DNA is really small, it can easily be degraded or contaminated. And basically what PCR does is it makes a bunch of copies of a specific region of DNA. And that's done by designing primers that basically tell a certain polymerase which area of the gene do we want to amplify, and then the polymerase amplifies that gene, makes a bunch of copies of it. And then in gel electrophoresis, we take all of those copies and run those copies through kind of a mesh. And gel electrophoresis is based on the fact that it's harder for a larger object to move through this mesh than it is for the smaller objects to move through that same mesh. And so these different DNA samples can be compared based on their sizes. They can be compared to a ladder that separates into bands of known sizes, and then the bands themselves can be compared as well. And all in all, this is based on the central dogma of molecular biology, that DNA goes to RNA, goes to protein. And in this experiment specifically, we use genomic DNA, gDNA for short, which has all of the introns and exons, which goes to RNA, which then we convert to cDNA because RNA is just really hard to work with. RNA is easily degraded. And so ultimately, expression is measured using cDNA.
And yeah, so as Justin was saying, we needed to go and design a primer specific to our WD40 gene. And as we can see here in the diagram, this is going to be the cDNA strand, meaning all the introns have already been spliced out. So this is just the coding region. If we look at these little triangles here, that's where our primer is going to be binding to our sequence. So we're looking at an expected length of around 581 base pairs. And then we went ahead and tested our primer. So we used RPPO as a positive control. RPPO is another gene found in our model organism. It's a housekeeping gene, meaning it's always going to be expressed, so we should always get results for the cDNA and gDNA lanes here. We use water as a negative control, and as we can see, both the water lanes for RPPO and WD40 have no bands in them as we expected. Our cDNA lanes for both RPPO and the WD40 do both have bands right where we're expecting them to, right around that 500 mark. And our gDNA lanes, unfortunately, the gDNA sample that we were using was most likely degraded. Didn't matter too much though, because we were focusing on that cDNA, and we went ahead and validated our primers. So we used them in the rest of our experiments.
And then going back to using PCR to measure levels of gene expression, we can also look at band intensity. And this is based on the fact that more expression will ultimately mean that more RNA is produced, and that will correlate with a more intense cDNA band on the gel. And just to reiterate, we're changing RNA into cDNA using something called reverse transcriptase, which just makes it possible to work with because RNA itself is just so hard to work with. But notice in this diagram on the right, these are two of the same gene under different conditions. And so on the left, there's a more intense cDNA band on the gel, which means more RNA was produced, and ultimately there was more expression. On the right, it's the opposite. There's a less intense cDNA band on the gel. There was less RNA and less expression.
So after we validated our primers, we moved forward with a similar study, but with an RT-PCR. And this introduces a whole bunch of controls, mainly that a minus RT sample will not have any other genomic material in the wells, and that just makes sure that there's no contamination from outside material that could interfere with our results. And from this, we created a PCR that had multiple primers, and we can just reference these all in one gel. And we constructed this by inducing DNA damage with hydroxyurea and untreated cells. And then this generated our cDNA that we then quantified. And then as you can see, we're just making the gel, checking our PCRs, and then loading the gel. And then starting out our PPO, we used that again just as a loading control, make sure that everything was run correctly. As you can see, there are no samples in any of the minus RT wells. And then down here at RAD51, that is just our baseline for DNA damage repair. As you can see in the HU well, it is more bright. So more cDNA samples are in those wells. That means it's more expressed because DNA damage was induced, and that just sets our baseline. And then looking at the WD-40 wells, it's kind of similar. The HU is definitely a bit brighter than the UT sample, and this we can then quantify in Fiji, which gives us a brightness level, which gives us our quantitative analysis. And this, we found that it was 1.3 times more expressed after DNA damage was induced than prior. And this is relatively similar to that of RAD51. And like all things in science, more studies need to be done, but we still believe that WD-40 is involved in the DNA damage repair pathway.
Yeah, so what's next? Chase just said, yep, we need further experimentation to confidently conclude whether or not WD-40 is playing a role in DNA damage repair. A couple experiments we could go ahead and do. Quantitative real-time PCR. Basically, it's going to be PCR, but you can analyze the expression levels in real time. We can also look at protein binding partners, things that aren't WD-40, but are related to the protein complex. That could also play a role in DNA damage repair. And then, yep, WD-40 is found in a bunch of different organisms. So being able to take that gene and put it in other organisms, that's also a step we could take in the future. We would like to thank our professor, Dr. Pinzone, as well as our great TA in LA, Carrie and Nishant, as well as the MCDB department at the University of Colorado Boulder, and the great Canadian person for the WD-40 flag. Thank you. Any questions?
We have one person raising their hand. Christopher, I'm gonna unmute you now. You can go ahead and ask your question.
Sorry, actually, this is just to take a hack at how many of the diagrams were up. I'm gonna guess it was 11, including the one you showed at the very beginning to show us what type it is.
That was a great guess. Christopher, I think you won our giveaway.
Yeah.
Yes. Thank you.
It was supposed to be when we asked for it, but
Oh, shit, I'm so
that's
sorry.
fine. No, you're good. You're good. Don't worry about
We
it.
know you're excited.
We're gonna we'll find a way. We'll write your name down and we'll find a way to get you our prize. We can show you the prize. Hang on. Yeah, we got
I'm
you your
sorry.
very own can of WD-40.
Oh, beautiful. Thank you.
Yeah. Which just we have to clarify is not related to the gene, but we just thought the name was kind of funny.
Not sponsored?
No.
Thank you. Okay. Ian has a question. I'm going to go ahead and unmute you. I think it's not wanting to do it right now. Let's just temporarily move over to the comment, to the question and answer section. There was a question about the loading control, and the question was related to whether or not the brighter band of the loading control had any significance in your data.
No, we thought it was just there was just more sample added into those wells. It's not directly an expression difference. It could be thought that after UT compared to HU that it decreases in expression because just the DNA is getting damaged and the gene's just not being expressed at that point as much as previous.
Yeah, but they're similar enough that the loading control we were able to conclude that was a good control.
Grace Patrick is asking in the question and answer section, do you have any guesses about the function of the protein product?
The protein products itself, I mean, we know that other WD-40 proteins are involved in DNA damage repair in some way. So they might not be directly involved, but they could also assist some other protein. That's what Alexi was talking about with using protein binding partners, which we could just look at what else is WD-40 protein combining with to assist in the potential DNA damage repair pathway. But then other than that, the WD-40 protein itself is involved in a lot of things. So it has a lot of different functions.
I think our last question that we have time for in the question and answer asks, how applicable do you think what we learn about its mechanisms of DNA repair will be to humans?
Yeah, I'd say since the gene is already found in humans, we can't say definitively, but should kind of follow the same rules that would follow in Tetrahymena thermophila. So we should be able to kind of take the gene from Tetrahymena thermophila and implant it in a human. Should see similar results.
And in general, DNA damage is a problem not only affecting humans, but it is a problem affecting a lot of things in life. Because DNA damage, if it's improperly repaired or if it's not repaired at all, can lead to cancer, which is a huge cause of death. And obviously, it's not something that we understand completely. And so DNA damage repair is something that we need to understand better to better understand cancer.
Great, thank you so much. People who have remaining questions in the question and answer area, just keep an eye there and someone will get to your question shortly.